finally, part three. We're going to cover part three and part four together in this next part. So evolutionary forces affect allele frequencies. So kind of what we're driving at is different populations have different allele frequencies. And so when we calculated the allele frequencies of different alleles, we found one had something like this, and one had a frequency that could be completely different. And so what are the reasons that different alleles have different frequencies in a population? And there's kind of two primary reasons for that. One is just kind of when they occur. So, if you look at like an A and a G that exists that's different within you, like that was the result of a single mutational event in some individual, hundreds of thousands of years ago. And so kind of, when these mutations occurred, did this occur like 50,000 years ago while this one occurred 200,000 years ago? Those can have very profound effects on the allele frequency, but then there's also these evolutionary forces that are responsible for changing them. So natural selection, genetic drift, migration, and bottlenecks are the four evolutionary forces we're going to cover. So natural selection, this is probably the most interesting type of change. So this is a major force driving allele frequency change. And it's a chief mechanism for transforming populations in terms of their phenotype. And so the idea is that there's a force that shifts allele frequencies within large populations that's dependent upon the phenotype of the individuals, and the phenotypes can make them more fit to their given environment. So this is originally discovered by Charles Darwin and independently by Alfred Wallace as kind of a mechanism that could explain how species change. So in this concept, individuals exhibit variations in phenotype. So kind of one of the things that Darwin talked about were these Darwin finches, where different birds would have different beak sizes, and that could impact whether or not they could eat certain seeds or not. And if these variations are heritable, controlled by genetic differences, then the individuals that have large beaks that allow these birds to eat and feed on particular seeds, their offspring might be more likely to survive in situations where those seeds are present. And so some phenotypes are more successful at survival and reproduce at higher rates. And so consequently, if individuals with a certain phenotype are more likely to survive, then those genes that they carry will also be changed at a similar frequency. So a human example of this is an example of positive selection in humans is lactase persistence. So we talked a little bit about this before. So milk has become an important part of humans' diet. So we drink milk from cows, from sheep, from goats. But this is very unusual if you look at like the mammalian lineage. So typically, only young offspring are able to drink milk. So because only offspring have typically drank milk, adult animals do not have the proper enzyme lactase in order to actually process that lactose sugar. And so recently humans have evolved the ability to metabolize milk as adults because there are genetic variants that change whether or not this lactase gene turns off as a function of developmental time. So, if you're expressing lactase, lactose is converted to glucose and galactose. This can be taken up by your small intestine and then metabolized. But if you don't have this lactase gene, then you end up having a large amount of lactose present, and so these can have effects on your digestive system, including the production of things like methane, and also osmotic shock, which causes things like diarrhea. And so this not only causes these side effects, but also kind of importantly, what's thought to be important for the spread of this lactase persistence is the fact that you can acquire the calories from cow's milk as an adult. And so, a large number of studies have identified alleles that are able to confer this phenotypic effect of lactase persistence. And so here are 5 different alleles that experimenters have evidence with these changes, these genetic changes. This is kind of like the location within the lactase gene and kind of the change. This position was changed to a G, but these are all different polymorphisms that are associated with the ability to produce lactase as adults and be able to metabolize milk as adults. And so you can actually see these different alleles have spread in different parts of the world. So there's alleles that have spread in Africa. There's alleles that have spread in Northern Europe. There's alleles that have spread in the Middle East. There's alleles that have spread in Sub-Saharan Africa. Etc. And so the idea is that this ability to drink milk has been beneficial in many different parts of the world, and different alleles have occurred and then been selected for. And so these alleles have spread through positive selection because individuals that were able to drink milk as adult were much more likely to survive, 20,000 years ago. Pathogens are also a very important part of natural selection. So Salmonella is an example of a pathogen that can infect humans, and there are alleles that have been identified that confer resistance to Salmonella. And so kind of you can see the distribution of this allele, this Salmonella resistance allele, and kind of where it's now commonly found in different parts of the world. And so this is related to kind of where this disease actually occurs in different parts of the world. So, this is just one of many different examples of how pathogens can play a very important role in natural selection and can end up increasing frequencies of alleles that confer some sort of resistance to that pathogen. EPAS1, so this is an adaptation to high altitude. So individuals that live in very high altitudes, one of the physiological changes is the amount of oxygen partial pressure. So the higher up you go, the less oxygen there actually is within the air. And so this is a very strong selective pressure because individuals have to be able to handle the fact that there's less oxygen when they breathe in for them to kind of like run their whole metabolism. And so one example of a genetic factor is EPAS1. So this is hypoxia-induced transcription factor, and this transcription factor controls a pathway that deals with low oxygen conditions. And so basically individuals that carry this allele kind of are preset to handle the lower oxygen levels of their normal environment. So you've kind of hijacked a pathway that is used for alternative, like kind of like rare events. So like if you're normally found in a place that has normal oxygen, you still have this pathway that's able to handle lower levels of oxygen, which is what hypoxia means. And so you kind of increase this pathway during normal life as a way to kind of handle living at higher environments. So this haplotype, if you compare these populations, so you're comparing Han Chinese from Beijing to Tibetan populations that live at these very high altitudes, and you can look at what are some of the alleles that are most different between these populations, and you can see one of the most extreme deviations is within this EPAS1 gene, which is how it was originally identified. Kind of interestingly, this allele actually comes from an ancestral human population, Denisovans. So it's thought that early mating between Denisovans and human populations, as they kind of as humans re-explored kind of these geographical areas, the humans could have picked up this haplotype from the Denisovan populations that already lived kind of within these. So these pre-adapted Denisovan populations already carried the alleles that allowed them to survive at these low oxygen levels, and then the humans were able to integrate that from them. So these are all examples of natural selection, and so these things, so if alleles affect phenotype, then these can be selected for, and subsequently you can change the frequency of an allele within a population. Migration, so other evolutionary forces that affect allele frequency are migration. So kind of one of the classical examples of this is looking at the Y chromosome. And so if a large number of people from one population migrate to another, they can bring high frequency alleles to these new populations. And so kind of the idea behind that can be seen with the Y chromosome that was carried by Genghis Khan. So because Genghis Khan ended up having so many offspring, because his offspring also kind of became leaders and had a large number of offsprings, there is this Y chromosome genotype, haplotype, that is very present in kind of the Mongolian ancestors of the Genghis Khan population. And so as they spread and kind of took over a large land area of mass, they ended up actually spreading this Y chromosome to other populations that normally don't carry it. And so this is an example of how migration can change the allele frequencies in other populations. And kind of one of the most important things, so it's kind of a boring evolutionary force, but this is probably the most important for just looking at, a given random allele in a population, is genetic drift. And so genetic drift is just chance, can have a very profound effect on changing the allele frequency from one generation to the other. And so if you have a starting population that is 50% one allele and 50% of another allele, each generation you're kind of randomly sampling of this in order to create the next generation. And so, we don't have infinite population. So this kind of stems from the fact that we don't have, this is why infinite population size is one of the assumptions of Hardy-Weinberg, is because if you don't draw an infinite number of alleles for the next generation, then random chance becomes very important. And so let's say you only had, 10, you had 50 offspring and you were selecting 5 for the next generation. And so even if the original frequency was 50-50, just by chance, there's a very high likelihood that it's not going to end up exactly 50-50. And so it might end up like 60-40. And so this change in frequency is permanent. So changing from 50-50 to 60-40, you're not going to go back to 50-50. There's no pressure to go back to 50-50. This is just kind of like a new starting point. And so, genetic drift can go both ways. So if you actually plot the allele frequency over time, it can go up and it can go down. But, over time, you're going to deviate from the original amount. And so this is kind of why this is a very important evolutionary force. And so if you have two populations that are separate from each other, even if they initially had that same allele frequency, just their particular random path will be different. And so they end up having these subtle differences in frequency or profound differences between them. Population effects are also bottlenecking, so a sharp reduction in the size of a population. This is also referred to as a founder effect. So if you have a bottleneck can either occur where you have a large number of people already living, but then there's some sort of catastrophic event where 95% of the people die and 5% are left to carry on and repopulate their current place. A founder effect occurs when a new environment that typically did not have individuals of a species are now founded. And typically what happens is that you either have, in both cases, you have a very small amount of individuals that actually seed that population. So that individual initial population can be very large and have a mixture of different alleles, but if you select a very small number through this bottleneck or through this founder, you can have very profound changes in allele frequencies because of that. And this can actually be seen in non-African populations. So because Africa was kind of the source of human evolution, that initial source, as populations left Africa to migrate to West Asia, to Europe, etc., it was actually a very small number of individuals, not small per se, but much smaller than what existed in Africa. And so because of that, there are these population bottlenecking and founder effects where the frequency and the number of alleles that are present in these European population and Asian populations is much smaller than what continues to exist in Africa today. All right, the final part is going to be talking about how we recognize familial relationships. So kind of as I said, the way Ancestry.com or 23andMe will recognize close relationships, it will be by looking for these kind of runs of DNA where the dye isn't random. So, if I looked within there, there'd be, hundreds of thousands of individual differences, individual SNPs. And so what we're looking for are what are called identity by descent. And so this idea is that, if you have two individuals that have the same parents, they are going to, this is showing you the paternal and maternal for the father, the paternal and the maternal for the mother. And so they are just going to inherit certain regions of the genome that are identical by descent. And so what do we mean by that? Well, so if you have a large number of offspring, all of the individuals that inherit this orange part of the chromosome are genetically identical to each other. And so these are runs of identical alleles. And so, again, if you compare two unrelated chromosomes, each one of these SNPs is kind of an independent die roll where, you don't expect having this allele to affect this allele. But when it comes to these identical by descent, what you're looking for is all of these SNPs between myself and my brother are exactly the same. And so there are these runs of identical SNPs that statistical approaches can identify. And so we can find regions of identity by descent that allows us to basically determine familial relationships. So for something like a brother, we expect approximately 50% of our genome to be identical by descent. And so some of that will be regions that are not identical at all. So in this region right here, I might have inherited the maternal chromosome from my mom and my dad, and he inherited the paternal chromosome from our mom and dad. Some of it, we share one of the two, so these are these light purple. And so in this case, we both inherited the maternal from my mom, but I inherited the paternal from my dad, and he inherited the maternal from my dad. And then you also have runs where it's identical to each other. And so these are areas where we inherited the same. So it could be something like maternal from my mom and then paternal from my dad, and we both inherited that same block. And so the reason these chromosomes, again, aren't identical to each other is because recombination creates hybrids. So each sperm has a unique location for kind of where these breakpoints occur. And then when you look, and so this is a important test, for example, to determine whether or not is this my brother or is this my half brother? And so the reason I know that this is my brother, my full brother, is because there's regions of the chromosomes where we're identical at both chromosome 1 and chromosome 2, and that's not possible for half brothers. And then second and third cousins, as you look at this relationship over time, the probability of these runs of identical descent, because you're sharing less and less direct ancestry with each other, become less probable. But you do end up seeing kind of these like smaller and smaller blocks. So these blocks, because they contain so many individual SNPs, are easy to identify, but they get smaller and smaller as recombination breaks it up more and more. And then finally, effects of inbreeding. So this is another consequence. This is another breakdown of Hardy-Weinberg. And so again, Hardy-Weinberg assumes that individuals that mate with each other are just kind of randomly sampled from the population. And so these are examples of this violation because individuals of this family were much more likely to marry and have children with other members of the family. And so this has a negative consequence because it can make disease alleles much more likely to end up as a homozygous state together. And so this would be a good kind of test question. So like, if you have a disease that has a very low frequency in the population, of 1/100th of 1%, kind of, in general, what that means is that the carrier frequency is low enough that, it's very unlikely that two individuals will come together that both are carriers. But because individuals of a family are much more likely, if one is a carrier, they're much more likely to also be a carrier, then you can end up having this violation of Hardy-Weinberg that causes a much higher frequency of homozygous recessive diseases. And then kind of this approach can also be used to infer ancestral inbreeding with non-modern humans. So because that allele frequency graph in Denisovans and Neanderthals is very different from the human allele frequency distribution, scientists have been able to actually recognize that in human evolution during the spread of humans out of Africa to different geographical parts of the world, they encountered these other human populations and bred with them and hybridized with them. And so we can now actually start to recognize parts of our DNA that came from Neanderthals or these Denisovans ancestral populations. And that has shed light on kind of human evolution and kind of some important hybridization events between modern humans and kind of these ancestral populations. And kind of one of the weird things that occurred, so one of the loci that are associated with COVID-19 outcomes, whether or not you have a bad or a positive outcome, this is also something that was actually identified and shown to be associated with Neanderthal DNA. All right, so that is chapter 26 lecture.